I welcome all the participants. And today I will talk about targeting mRNA conformational ensemble for developing therapeutics for SMA. SMA is spinal muscular atrophy, is a genetic disease, and we will talk about that in a moment. And the two questions that will be addressed during this talk are the following. Can we target the conformational ensemble of RNA structure with small molecules and modify the splicing to revert the pathological phenotype? This was the, the question we had when we start this project. And of course, is this type of intervention, does it have a venue to go to clinic? Is there any other examples with small molecule modifying splicing that went into the clinic? And we will end up then with a conclusion. Spinal muscular atrophy, as I told, is a rare disease, is a neuromuscular disorder characterized by the loss of the motor neuron, as you can see here. I will try some things to have the laser pointer. Is it okay? You see it?
Yes.
Okay. So this is a normal motor neuron. This is a motor neuron of a SMA patient. And because the motor neurons are not functioning, these go down then to the muscles. This is a normal muscle, and this is a SMA muscle. It doesn't touch only muscles, but joints, but breath, the lungs, so you have breathing difficulty, the heart. So it's a disease that really impairs many functions of a human being. The next slide, I have to see, okay, I have to manage it like that, shows you that SMA has different type of disease. And you should keep that in mind. So the more severe one is 0, the less severe one is 4, and the difference between this type is due to the number of copy of one gene that we call now the SMN2 gene. Okay? There is a correlation between the degree of severity and the copy of this SMN2 gene. But why? Because the disease is caused by the fact that one other gene called SMN1 gene is not working anymore. So normally, everyone has a SMN1 gene and a SMN2 gene. The SMN1 gene has the right splicing and produce the right protein, which is the survival motor neuron protein, SMN for survival motor neuron. And the survival motor neuron protein is a splicing protein per se. The SMN2 gene has the same exon, the same intron, besides one mutation on exon 7. The C, the cytidine, is changed in a uridine. And this does not allow anymore the splicing to be full. So you have the creation of proteins missing the E7 exon, and thus they are truncated, non-functional. And this SMN2 gene is able to produce only 10% of the survival motor neuron protein. So when a patient has more copy of the SMN2 gene, he will produce more SMN protein, and the severity of the disease is lower. This was known for many years, and now when you have the disease, the SMN1 does not work, and the only protein that is produced is the one of SMN2. If there is too less, the severity of the disease is higher. So, how to go about solving the problem? Having a therapeutic that works, disease modifying. So we know the more the more copies of SMN2 are present, the better the disease or the less severe the disease is. So one approach has been to increase the expression of SMN2. Small molecules have been used in animal, in vivo, proof of concept, and in man, but they were not so successful. Most successful was the idea of changing the splicing of SMN2. So here you have the DNA, and that's why you have a T instead of the U. And this has been achieved. The first proof of concept has been done with an antisense oligonucleotide. This was also the first antisense oligonucleotide working. It's called Spinraza, or the compound name is Nusinersen. How does this work? What is the principle? Is shown here. This antisense block the intron splicing silencer, and thereof increase the exon 7 splicing and incorporation, and thereof increase the SMN protein expression. That's how it works. So in the figure you have here the normal patient, the normal person with no mutation in the SMN gene, and here you have the situation in the patient, and that's where this Nusinersen function. But this is a quite complicated intervention. This has to be given intrathecally. It's quite complicated, and right now also quite costly. It's cost about 1 million for a treatment. So people were still working on small molecule. Is it possible to change the splicing of SMN2 using small molecules? And in 2014 and 2015, Roche and Novartis came out with very nice paper showing that it is possible to modulate SMN2 splicing with small molecule by tackling the U1 pre-mRNA association, and that this rescues SMA mice. So a clear mechanism of action and a clear effect in vivo. Very good news for the patient, and we will see at the end how this was pushed forward towards the clinic. Our question was, because at the beginning those compounds had quite a lot of problem linked even to the mechanism of action, problem in sense of toxicity, was, is it possible to target directly the mRNA and influence the splicing by targeting the mRNA? An analysis of the exon intron situation of SMN2 gene showed us that there is an hairpin called TSL2 that may be very important for the splicing in the case of SMN2 gene, in particular for including the exon 7. Why this is important? Because this hairpin is just containing the 5' splicing site, where the U1 snRNP is coming, and so the machinery is starting. And our idea was that this hairpin could have multiple conformations. And one of these conformations is able to modulate correctly the splicing. And that's why 10% of SMN protein is produced by the SMN2 gene. Whereas the mutation that impairs the SMN2 gene to be spliced completely is the one that impairs the binding of the enhancer splicing element that promote the inclusion of the exon 7. So it's here we are tackling a structure that is just at the beginning of the spliceosome, or is just the one interacting with the spliceosome. So we ask the question, does TSL2, this hairpin, have different conformation? So can we map the conformational ensemble? And to do that, we did a mini gene experiment trying to see how exon 7 is spliced corrected, incorporated. You see here SMN1, this is m-RNA, and you see the incorporation of E7. And in the case of SMN2, you see that this is not incorporated. You see only a small band. Then we ask the question, what happens if we mutate the hairpin? Are we able to increase the splicing and increase the inclusion of E7? In fact, you have to think at the hairpin as your arm and your hands bound together and doing a scissor movement. So what we speculate was, or we asked the question, can we transform SMN1 that incorporate E7 into SMN2 by mutating one base pair? And that's what we did. Here you see a mismatch. So we substitute the uracil with a cytosine, and we make a 3 hydrogen bond, so we strengthen the stem. And what happens? Our SMN1 gene modified is not able anymore to splice. Oh, interesting. Now, making something worse is always easy in science. Can we do the reverse? Can we introduce a mutation in SMN2 and increase the splicing? So we decided to add a mismatch to loosen the stem. And here an example, the 3C, and you see that the splicing is ameliorating. So there is more E7 incorporated. And for the rest of the mutation, we had the same proof of concept. So this is at the level of the mRNA. At the biophysical level, we did an experiment with 2-azopurine. When the 2-azopurine are close to the rest of the stem, there is no fluorescence. When it's open, there is fluorescence. And you see there is a correlation of the E7 inclusion with the 2-azopurine emission. We have one outlier, this is force G. We could not yet understand it really why. The same with gels showing different structure. So the hairpin content is inversely proportional to the E7 inclusion. The more we have of the hairpin, the less inclusion we have. And the less hairpin, the more inclusion we have. And the same we have it with circular decrease. So this full slide with a lot of experiment show us that TSL2 has a conformational ensemble that can be modified, or it suggests at least a conformational ensemble that can be modified. And this modification can modulate the splicing. So the idea and the plan was, can we find a small molecule that allow us to make the 5' splicing site accessible or more accessible to the spliceosome. For that, we went to chemistry. Was a publication showing molecule or chemical groups that likes to bind mRNA. We use them to filter a library that we had in-house from a previous project, European project. And we use for the screening an assay where we use displacement. So a fluorophore to Pro was added to the mRNA, and if a ligand was displacing this fluorophore, the fluorescence was reduced. And you see there were some compounds being able to displace the fluorophore. Here you have a dose response curve and the structure of this compound. And then we went ahead and asked ourselves whether this first target-based screening of SMA can be translated in results of splicing into cells. But before doing that, we were asking, does it really bind to TSL2? And this was a very difficult task because the solubility of this compound, PK49 in particular, was not good. So with ITC, we saw a first hint of binding with the grating coupled interferometry signal by Triloop. We saw that there is binding when you increase the concentration, but in both cases we could not reach the KD value, so we have only an estimate of the KD that is around 100 micromolar. Whereas we had an effect at 12 micromolar within the displacement experiment. We went into cells, two systems, a mini gene system based on HeLa cells transduced with the mini genes carrying E6, E7, E8, and then the readout real-time PCR and the mRNA. On the other end, we went into patient cells. We had the cell line 14B. This is the parental cell line. The two parents have still one SMN1 gene, so producing sufficient SMN protein, and their child had no SMN1, functioning SMN1 and only SMN2. So it's the disease model. These are the 13C cell line. So we went with this compound displacing the fluorophore, and you see here that starting from 10 micromolar PK49 is able to modulate the splicing and have a complete incorporation of E7. I focus on PK49 because indeed its toxicity is low. There is no toxicity at the concentration where we see an effect. Whereas the other compound had higher toxicity, and thereof eventually the splicing effect that you see may be biased by the toxicity or the general toxicity. So we went further with PK49. We went into the patient cells, and we were very happy to see that even in patient cells, the compound is able to modulate the splicing, and the E7 exon is completely integrated. Thereof, the next question was what happens in vivo. So we use a Drosophila model, which is a splicing model, is not a disease model. And also there, although the bands are faint, at higher concentration, of the compound into the water given ad libitum to the Drosophila, we have an amelioration of the splicing and of the incorporation of the E7 exon. This is all fine, but this is mRNA. The subsequent question was, what about the protein? Do we have more protein? You have here the Western blot. This is the situation. The patient, 13C, the parent. Of course, the parent expresses more SMN protein than the son, the patient. And here you see what happens when you treat the 13C cell with the compound. So you have an increase of SMN protein. When you do it and you observe it with the microscope, SMN protein as a characteristic is to form gems within the nucleus. And these are the gems. So untreated, there are few gems at this point. When you treat them, the number of gems increased. Okay, we have an effect on cells, on patient cells. We could translate the mini gene to the patient cells. We could translate the in vitro targeted assay into results in positive results in patient cells. But what about the selectivity of this compound? And the good things in targeting mRNA is that you can always go back and do RNAseq to answer the question of specificity. So RNAseq has been used many times within the spinal muscular atrophy field for distinguishing the effect of compound, the mutations, and so on and so on. And we did it. We did it with the help of the bioinformatic group of Roche. So they help us in the analysis. And what is interesting is that we were expecting 1,000 genes to be modified. If we tell the truth, that was the expectation. In contrast, at the end of the analysis, we saw that 201 genes, including SMN2, were spliced or their splicing was modified. And we treated the cells for 6 hours, in contrast to the work done by Roche and Novartis, where they had a very short treatment to see the first event. So in our case, SMN protein, that is a splicing protein, was already present. So there are several spliced genes that are spliced because SMN was present, the protein was present. The gene set enrichment analysis returned us a very interesting data showing that indeed there is no overrepresented biological pathway, indicating that this splicing corresponds to a mechanistic effect rather than a concerted toxic response. So, to the question, is it specific? The answer is not bad for the stage of the compound being validated it. You can do better, but in fact, what counts is which of the protein or of the gene of the mRNA are really spliced. And we could clearly show that there are mRNA spliced because of the production of SMN protein. You see it here. This is the disease model cell line. The 14B, the parents that have already sufficient SMN protein, and a cell line that has no problem with SMN genes, neither SMN1 nor SMN2, and you see only the one of the patient respond in all these 3 cases. Whereas we know it, there are other genes where the relationship is clear. There is an off-target effect because also the cells, normal cells, react on the compound. Now, we have shown that there is a binding, but the question here is where it's bind. And I have told you that the compound has a problem, the solubility. So I can ensure you already that we cannot do the NMR structure with the compound because of the solubility. But we could study the binding motif on the TSL2, and there is this box, which is the putative binding model for motif for such a compound. This is what you find also in TSL2. We analyze what is the effect of the compound in the different mutant that you saw at the beginning, and in principle we saw always an effect when the mutant was having less E7 incorporated without treatment, and then with the treatment the incorporation was higher. And one mutant, the 3G70C, that was made in order that the stem is so rigid that you cannot open it anymore, and in this case even the compound activity was not enough to make this mutant splicing properly. So if the stem is too tight, then you cannot have any splicing. Saying like that, we had a hint where it could bind, but the analysis of the NMR analysis of the compound with the TSL2 hairpin show clearly that you have modification of the signal for the base U2 and U19. Thereof, most likely the compound is interacting with these 2 base pair. So we focus on that, and because we could get the structure of TSL2 alone, you see it here, and this is the complete bundle. What was interesting was that we found in this bundle 2 distinct conformations. The one that we call pentaloop, with a larger loop here, and the one that we call Triloop, with a shorter loop, as depicted. This is the penta, this is the 3. 80% of the structure in this bundle are penta, and 20 are Triloop. Is there any hint? The Triloop is the one fostering the splicing. The penta is the one, as we depicted here, is the one blocking the access of the 5' splicing site to the spliceosome. This was a question he had, and we wanted to address. And that's where we switched to molecular modeling, because we could not get the structure. And we did docking also with the Biosolvit software. So we used many different solvent docking program in order to see what was the consensus, and all had a consensus that the molecule would bind between U2 and U19, and that was confirmed by the NMR analysis I showed you before. So in tempore, we did first the modeling, and then we had the NMR analysis. But here in the presentation, I presented the other way around. And what happens if you do molecular dynamic with this system? This is the penta conformation without the ligand and with the ligand. And as you can see, with the ligand, the distance between A1 to U90 is increased. So it's like you go from this situation, this conformation, to this one. And what happened above in the loop, here and here, by binding the compound at the bottom, you see it here. The penta without the ligand, the distance between A8 and U12. So we are talking about the position here above is big when you add the compound, so it gets shorter. So it looks really like the compound is able to shift from the penta to the Triloop. We went back to our mutant. We did the analysis with molecular dynamic simulation. Also here, 40 nanoseconds were enough to see the effect. We carried it longer for some of them, but the image did not change. And indeed, 2A, which is splicing more, has a longer distance between A1 and U19. We are talking about here, so it's open. It's presenting the 5' splicing site. The 3G and 17C, so this is the one where we strengthen even more the stem. The distance is very, very short. So the 5' is sequestered in this situation where we have a clear pentaloop. And experimentally, we decided to force the Triloop. So what did we do? Here above you see it's A8U12. So 2 between adenosine and uridine, you have 2 Watson-Crick hydrogen bonds. And we decided to strengthen it. We changed the 2 base pair and we did a GC base pair where we have 3 hydrogen bonds. So we forced a Triloop, and this mutant AG12C clearly show an amazing insertion of the E7 exon. So indirectly pointing that there is a correlation again with between the Triloop conformation and the presentation of the 5' splicing site. The more Triloop conformation, the more the 5' splicing site is presented, and the better is the incorporation of E7. So we have this correlation between the Triloop and the splicing. So what, in summary, we are almost at the end. What we got out of this exercise and this study, that TSL2 is a suitable target, that small molecule screening works, and we could identify 54 hits. Because we were looking directly at the SMN RNA, we had a good translation between the in vitro with the isolated mRNA and the HeLa cell into the patient cells, and PK49 is a promising hit to be developed further, and we are working on the synthesis of derivatives with our collaborator in Lyon. And that PK49 binds to TSL2 to this motif of the exon 7 5' splicing site, and it's open temporary the hairpin, allowing then a better docking of the splices. What does our work add to the field? We showed that it's possible to do target-based small molecule screening of SMA and eventually example for other splicing-linked disease. We showed that the mini-gene technology can translate into patient if you do a direct measurement of the E7 RNA inclusion via RT-PCR. We had a long discussion with Roche, and they had a lot of trouble in translating their mini-gene into patient cells results, because the mini-gene they were using reporter gene, and they were amazed when they saw our data where the translation was very, very good. We have now deposited the first NMR structure of TSL2, and the rationale for drug discovery is there that if we can manage the shift from penta to Triloop of TSL2, we can modify and ameliorate modulate the splicing in SMN2 and then revert the phenotype. Okay, I was telling you, and this is what we did, but what did other people do? They worked a lot, and Roche in particular. So they published recently this paper where they describe risdiplam. Risdiplam is an evolution of the molecule shown in the science paper in 2014, and risdiplam is now in clinical trial. So it's a modulator of SMN2 splicing using another mechanism of action compared to the targeting directly of the m-RNA, but the results for the patient are very, very good. There is a sustained increase of SMN protein across type 1, 2, and 3 SMA. There is an improved motor function in babies with type 1 SMA, and we expect, based on the results shown or seen in the animal model, that the increase in SMN protein will lead to significant clinical benefits. So it's ongoing, and before I conclude, there is also a gene therapy approach that is ongoing with AAV vector, and it seems also to work nicely. The only problem is the dosage is so high that it takes 1 year to produce one single dose for one patient. The cost is very high, and right now these are limiting factors. And furthermore, the possibility of having a reaction, an immune reaction, makes the gene therapy being a one-shot therapy. If it works, it works. If not, you cannot really repeat it. And thereof, in the field, everyone thinks that a combination gene therapy plus pharmacological intervention with small molecule is the way to go. But to conclude, I would say that SMN2 is small molecule splicing modulator with different molecular activity mechanism of action represent a real disease modifying therapy. So to go from this situation where there is only a small production of correct spliced and correct SMN protein, to this situation where splicing is complete, SMN is produced, and you see here the gem of this SMN. And with that, I'm finished. I would thank the people working on this paper, particularly Amparo Garcia Lopez, who did most of the work, Francesca Tesero, did all the molecular modeling, and dynamic work was at a given moment in close collaboration with Modesto, Roscoe, and Faustino. For the NMR, we relied on Professor Schwalbe's group, and in particular Anna Wacker. The compound came from the collaboration with the University of Lyon. For the bioinformatics, we could count on a nice collaboration with the people of Hoffmann La Roche. And for the Drosophila experiment, I thanked Ruben Artero for the collaboration. Here you see the sponsoring, and the rest of the group is shown here with the different sponsoring of the group. And I thank you them for all the work they are doing, also in other projects, and you for the attention.